In this video, we are going to be learning about the role and structure of lipids. Lipids are useful as energy storage, insulation in nerve cells, protection around organs, and as hormones. There are two main lipids that you need to know about. The first one is triglycerides. These are fats that are made up of one glycerol molecule and 3 fatty acid chains bonded to the glycerol. Glycerol is a free carbon molecule in which all the carbons are attached to an OH alcohol group. The rest of the bonds are with hydrogen. A fatty acid is a long chain of carbon atoms with one alcohol OH group. When the two OH groups come together, a water molecule is given off and an ester bond is formed. Triglycerides are used as storage molecules. The fatty acids can be of two different types: saturated fatty acids and unsaturated fatty acids. Saturated fatty acids are those in which there are no double bonds between carbon atoms. Unsaturated fatty acids have at least one carbon-carbon double bond. The bonding of saturated or unsaturated fatty acids to the glycerol is important. Saturated fatty acids have straight chains, so if only saturated fatty acids have bonded to the glycerol, the density of the triglyceride is higher as all the molecules fit into a smaller volume. This is why saturated fatty acids are usually solid at room temperature. Unsaturated fatty acids have carbon-carbon double bonds. These double bonds make the fatty acid chains bent. As these fatty acids bond with the glycerol molecule, they take up more space and are therefore less dense. This is why unsaturated fats are usually liquid at room temperature. The other fat molecule you need to know about are phospholipids. Phospholipids are found in the cell surface membrane of cells. Phospholipids are made up of glycerol that are attached to two fatty acid chains. The third bond that glycerol makes is with a phosphate group. This changes the properties of the phospholipid and makes it different from the properties of the triglycerides. Where triglycerides are insoluble in water, phospholipids have a hydrophilic head, which means the head likes water. But phospholipids have a hydrophobic tail, meaning the tail dislikes water. To test for lipids, we carry out the Emulsion Test. For this test, there are 5 steps. The first step is that you take a dry, grease-free test tube. Step number 2 is that you place 20 centimeters cubed of the sample and about 5 centimeters cubed of ethanol. You then shake this thoroughly to dissolve any lipids. You then add 5 centimeters cubed of water and shake the test tube again. A white color indicates the presence of a lipid. An exam tip is to write white, do not write cloudy. The key points are: triglyceride are fats that are made up of one glycerol molecule and 3 fatty acid chains bonded to the glycerol. Fatty acids join with glycerol through ester bonds. There are 2 different types of fatty acids: saturated fatty acids and unsaturated fatty acids. Phospholipids are made up of glycerol that are attached to 2 fatty acid chains. Phospholipids have a hydrophilic head and a hydrophobic tail.